So welcome everyone. Today we're going to utilize the QIAGEN OmicSoft Studio to take a look at how you can use this to analyze your expression data. And so before we get started, I just want to let you know that the product that I'll be showing today is intended for molecular biology applications only. This is not intended for the diagnosis, prevention, or treatment of a disease. Now, today's session has some cool features, and I am going to try to make it interactive just to get some feedback in terms of, how I'm doing, what I'm presenting, is it relevant to you? So at the bottom of your screen, you're going to see this Q&A box. This is going to allow you to type in your questions. So any questions you have, please go ahead and use that Q&A box so that it just allows us to keep track of it a little bit better. Now, you're also going to see a raise hands button. And so if you guys can go ahead and just click on that button just to make sure that it works for those of you attending today, that would be awesome. So this is kind of like a way that I'll utilize to get some feedback in terms of whether you're liking what we are presenting today. So I can see that the raise hand button works for many of you. And then lastly, if you have any technical questions, please let us know through chat, and we'll try to help you as best as we can. But if you do have questions, please utilize that Q&A box. It makes things a lot more easier. And so today we will look at how you can use OmicSoft Studio to analyze your expression data. And so we'll start off by going over how to create a project and import your data. Now, many of you have different starting points. Some of you may be dealing with raw FASTQ files from the sequencer, while some of you may already have an expression matrix. And so using bulk RNA-Seq as our example for today, we'll go over how you can process the FASTQ files using a pipeline to generate gene or transcript counts. And then we'll look at how you can import an expression matrix that you obtain outside of OmicSoft. And also how you can import your annotation information as well. And so what we're going to do is we'll then perform some statistical analysis and take a look at some visual outputs such as PCA plots, volcano plots, and heat maps generated from your input data. And then lastly, we'll look at how you can gain additional insights. And so here I'll highlight how you can import your results from your statistical analysis into QIAGEN's genomic pathway analysis for biological interpretation, and how you can leverage the analyzed public data that's available through the OmicSoft Lands platform to compare what is biologically similar or dissimilar to the findings in your own data set. And so I did provide some step-by-step instructions for today's session that you can easily access by just utilizing any of these links. So what I'm going to do is I'm going to go ahead and paste these links in the chat box so that you guys can have access to them. Now, these links are also included in the slides that Kristen just posted. I was kind of like a little cheat sheet to get there a little bit faster. And so for today, let's imagine that your goal is to utilize OmicSoft to analyze expression data coming from RNA-Seq. And so using a very simple analytical pipeline within OmicSoft Studio, you can upload your FASTQ files to generate a count matrix with expression values per gene, per sample, based on your sequencing data, or you can import your matrix file if you are starting off your analysis using a matrix. Now, whether you import or generate a count matrix within OmicSoft, you can use the matrices with a gene level information to do comparisons between groups and calculate differential expression with some fold changes and some p-values. And so here in this example, we are comparing two cell lines and have fold changes and p-values for a set of genes that are differentially expressed between the cell line 1 and cell line 2. Now, many users like you want to capture what are the sources of variability within their data. And so for that, you can easily generate a PCA plot to look for clusters to see how your data clusters and then identify any outliers that may be present within your data set. Now, a common output many users like you want to look at is a volcano plot of their expression data. And so this gives you an overall view of how genes are either going to be upregulated in your data or downregulated in your data between your groups. And so here we can identify significantly upregulated genes by taking a look at the right-hand side, and significantly downregulated genes by taking a look at the left-hand side between your experimental conditions. And so here, for example, we can see that CCND1, which is an important promoter of cell cycle, is highly expressed in breast cell line MCF7 compared to bone marrow cell line K562. And so many users like you will utilize this output to generate a list of significantly differentially expressed genes, and using that list, you can then take a look at it as a hierarchical clustering in the form of a heat map. And so by generating a heat map, you can see how your genes are expressed across your different samples or your different experimental groups. So here on the x-axis, you have grouping of your different samples, and on the y-axis, you have a grouping of the different genes. And so colors represent expression, where red is high expression, and then here the green represents low expression. And you can see, for example, that this set of genes is highly expressed. Taking a look at the bone marrow tissue, whereas if you take a look at the breast tissue, they are, downregulated. So this allows you to capture some unique genes that are expressed in one group versus the other. And so based on the pictures and illustrations that I have shown you so far, just to get some feedback, are these some of the graphics or illustrations that you would be interested in by using the OmicSoft Studio, and if you are working with bulk RNA-Seq. So if you guys are able just to fill in that poll that just popped into your screen, just giving us some feedback in terms of, the illustrations that I've shown and the ones that we're going to be generating. So thanks everyone for participating in the poll. All right, so just a raise of hands, how many of you are familiar with the RNA-Seq pipeline? Just to get some feedback in terms of your knowledge. So I can see that some of you are familiar with the RNA-Seq pipeline, which is great. And so basically, for those of you that are not, a pipeline for RNA-Seq analysis typically consists of mapping sequencing reads to your reference genome, and then it involves performing some QC analysis, quantifying the expression levels of genes and transcripts, performing a differential analysis of gene expression among different biological conditions, and then performing biological interpretation of differentially expressed genes using pathway analysis or a gene set analysis. And so today you will learn how to perform these RNA-Seq analysis steps using a simple and very easy to use pipeline by starting with your raw FASTQ files, since many of you are interested in that. And then we'll also look at how you can utilize your count matrix as a starting point, since many of you are also interested in that as well. And so I want to point out that OmicSoft has tools pre-built for both gene set analysis and pathway analysis using engineering pathway analysis, and we'll go over that later today. Now, what are we going to be looking at today? And so for today, we are going to use a very simple data set looking at two groups of samples coming from breast and bone marrow cell lines. And so, we're using a simple set today, but just do note that all of the steps that we'll be going over can be used for much more complex, or if you are working more different groups. Okay. And so just to kind of remind everyone that today's data that we'll be looking at from a matrix standpoint is available for you to download and access. So if you do have a copy of the slides, you can go ahead and check out, this slide and download those individual files by simply just clicking on the hyperlinks and then either choosing to download from the left panel or just hovering over the document itself and then downloading from the bottom. So either way, these are available for you to use. Okay, so let's go ahead and get started. So for those of you that are very new to OmicSoft Studio, in OmicSoft Studio, analysis is going to be done under a project. And this can be done locally or on a server. So a local project is going to be saved on your local computer and runs the analysis on your local computer. There are two types of local projects. We have a simple and a distributed project. So in a simple project, all outputs are saved in a single folder on your computer and is generally recommended for microarray or RT-PCR projects, for example. Now, in a distributed project, the outputs are saved in separate folders, or actually separate subfolders, which are based on the organization of the data being analyzed and are generally recommended for larger projects like NGS data. And so today we'll be doing, give you an example of those. Now, in addition to doing local projects, you can also do a server project. And so a server project is a distributed project saved on the server and runs the analysis on the server. And so unlike a local project, a server project has required metadata fields that you do need to fill out to complete and create a project. And these fields are normally assigned by your administrator. So these are going to be the ones that have little stars, right? And so server projects are recommended when dealing with larger data sets, as they usually require more computing power. And so how would you go about to do this? And so to create a server project, you would have to log into your server. So when you open up OmicSoft, you'll go into that server tab, and then you'll enter your credentials, and you will sign in. And so once you sign in, you're able to create that server project. And I'll go over that once we go into the software. Now, although the steps for creating local projects are different than from creating a server project, all project types share the same structure. And so regardless of how the project is created, a new project will contain slots for folders. It's going to contain the slots for lists. It's going to contain slots for text and attachments. So it will use the same tools for processing and for JavaScript analysis, right? So even though we're looking at a local, distributed, single file, or a project, you can see that it has the same contents within it. So there's really not much difference there. And so one last time before we go into the software, step-by-step instructions for today's session can be accessed by clicking on either of these links. So if you guys are interested in starting out from a local analysis, you can check out this link. Or if you're interested in starting from a server analysis, you can do so. And once again, I will go ahead and post that information just so that you guys are aware of it. Okay, and so let's go ahead and go into the software. So for those of you that are new, this is what the OmicSoft GUI looks like. So here on the upper left-hand side, we have some tabs. So for those of you that are familiar with OmicSoft, many of you are probably familiar with our Lands tab. This is what we have, the public data that we have processed for hundreds of thousands of samples coming from sources such as Geo, SRA, and many more. Now, today we're actually going to be focusing on the Analysis tab. And so the Analysis tab allows you to perform a lot of analytical operations within the OmicSoft Studio platform. And so today we'll be working in the Analysis tab of OmicSoft Studio to perform our RNA-Seq analysis. Now, taking a look at that Analysis tab, you're going to see a screen that kind of looks a little bit like what you're seeing on my screen. And so here on the left-hand side, we have the Solution Explorer. And so this is the directory that contains all of the files you import and all of the outputs you generate using your data. On the right-hand side, we have the View controller. And so this will contain different options to help you customize your outputs. Now, to start the RNA-Seq analysis, the first step is to create a project that will store your input and output files by simply just going to the New option. And so here under New, you have the option to create a local project, or you have the option to create a server project. And so in OmicSoft, analysis is done under a project, right, and can be done locally or on a server. Local projects use the resources on your computer to run most of the tools that are available for NGS analysis. And then a server project requires that you connect to a server. The analysis is set up on your desktop or your local computer, and then it goes to the queue to be run on the servers. So if you have a small project that's not going to take a lot of your computing power, then you can use a local project. Or if you're going to be working with multiple projects that require a lot of computing power, then you can use a server project. And so for today, I'm going to just quickly highlight how you can start either. So if you're interested in doing a server project, again, you do have to be logged in to the server, right? So one way to check is by simply going to your server tab and then making sure that you're logged in. So if you're not logged in, so let's say that I wasn't logged in, go ahead. Then this is what your login would look like. So here you would just enter your credentials and then sign in, and that's going to automatically sign you on to the server. And so once you have that, you can simply go back to the analysis tab and create your server project. And to create a server project, you can just simply click on New and then Server Project, and then you'll see a window that will open up, allowing you to enter that required information that is needed, set by your institution to create that project. So in this case, our only requirement is that in my case, is that I have a title. So I'll just put demo folder and then create. Oops, sorry, also forgot that. And so that's going to create a server project that you can easily just use for your analysis. Now, one of the things to note is that when you are working with the server project, any files that you are going to be utilizing, they do have to be on the server first. So if you go here to the server tab and you go under the server files and browse files, your files that you're going to be utilizing for the analysis do have to be uploaded onto the server, so you can easily upload your files by simply going to the upload and then selecting your files locally on your computer. So once you have uploaded your files, those files can easily be utilized for the analysis within the server project. Now for today, we are actually going to be focusing and working on a local project. So if we go to the new, and then here we select the local project, this is going to give you two options. So a distributed project creates a subfolder for each file you create, and then a simple project puts everything in one folder in your local computer. And so today, because we are working with a very simple data set, we'll select a simple project. And so here, we just select the type of project that you're interested in. And then what you're going to do is you're going to go ahead and browse where you want to save that project. So in this case, when we click on Browse, we can specify where we want to save that information. So let's say that here on my desktop, I actually want to save it under my folder called Local Projects. And then I can just select the project that I'm going to, or name the project that I'm going to utilize. So now that you've selected where you want to save that information, you can simply just click on OK. And that is going to create a local project for you to utilize. So just to kind of review, you do have two options, a local project and a server project. Local projects does the analysis locally on your computer. A server project does require you to log in first to the server, upload your files to the server, and then create that server project. All right. And so with this, let's go ahead and look at how we can analyze our RNA-Seq data. So to start the RNA-Seq analysis from FASTQ files, you're going to use you can use a ready-to-use pipeline that allows you to perform the analysis in one step, or you can do things step by step. So here under the Solution Explorer, the tabs in the bottom, you can see that we have two different tabs that show up. So one way is to go in here and take a look at our workflow. So our workflow allows you to do the analysis step by step. So you can create the new folder, which is the first step, and then you can, for example, map your reads. You can do some pre-processing, some quality control, and so forth. So if you like to be a little bit more hands-on and interactive in terms of the different parameters that are going to be utilized for the analysis, you can do things step by step by using the workflow. But if you are more open to the idea of doing your analysis in one easy step, then the best thing to do is to run a pipeline to do your analysis. And so to do that, you would simply go to the option Add Data, and then Add NGS Data, and then here Add from a pipeline, and you can see that we have a RNA-Seq pipeline ready for you. So once you select the RNA-Seq pipeline, that is going to open up a window that allows you to set up your analysis. And so here we can use this option to add our FASTQ files, right? So if this was a server project, right, those FASTQ files would be first uploaded onto the server. Now this is a local project, right? So if you click on the Add option, we can go and take a look at your folders locally to decide, where you have that data saved. So in this case, I can see that we have some of our folders located, some of our data located here. You just simply select your files, open to import your FASTQ files. So once you have your FASTQ files, if you are working with pair reads, which is the case here, you do want to make sure that the pair reads are listed. Next would be to select your genome. And so here in the basic section, you can select the genome reference that's going to be used to map your reads. And so we do have different references that are available for you to choose from. And so here, for example, this particular data set uses this particular reference. And so we can go ahead and select that. But if you don't have, if you don't see your specific reference of interest, let us know and we can add it here for you. The next thing would be to select your gene model because you do want to quantify the expression at gene level. And so here we do have different options to take a look at what gene model you want to utilize. For this particular sample, this is the gene model that I'm going to use. But if you don't find the gene model that you're interested in, you can always contact our support team and we will help you add your gene model of interest here. Now, if we go into the reporting section, you can select the different types of outputs that you are interested in generating from your data. So if you want to QC your FASTQ files, you can select the option to perform raw data QC. Typically, users like you will perform the quantification step, which is already pre-selected for you. This will allow you to generate an expression matrix from your FASTQ files. This section also provides you with additional options. So for example, some of you may be interested in identifying fusions and mutations information. So you can go ahead and select those options. Or if you're not, you can just deselect them if you're not interested in generating those different outputs. So once you select the outputs that you are interested in generating, the last thing to do is just to name your output and then select where locally do you want to save that information. So once you select a folder that you're interested in, then the last option is going to be to submit that analysis. So I've already done this ahead of time, so I'm not going to go ahead and click submit. But just know that if you do have any questions about this, the help menu, the help option will open up the browser and will open to that particular section where it will guide you in terms of what the different operations in this pipeline are, and will give you some guidelines in terms of what files to add and so forth. So again, any help that you need to better understand, you can always click on the help. So I'm going to go ahead and just click on cancel, just because this was done ahead of time. But by using this pipeline, just like you can easily analyze the RNA-Seq data in one easy step, where you're going to QC, map your read to the reference, and then generate some gene and transcript counts. Now, for those of you that are a little bit more hands-on, you can always use the workflow option to run those individual steps. Now, once the pipeline is done, it is going to generate outputs for raw and normalized gene and transcript counts and various types of reports. So here, just to show you an example, it's going to provide you with information on your NGS data, letting you know what data was ran. So here is your information on your files. That NGS data is going to be then generating a Omics data or process data where we have your counts as well as your normalized information. And then it's also going to generate different types of reports. So it's going to do a QC report and then some raw data and so forth, depending on what you selected as those outputs. And so this allows you to easily analyze your FASTQ files from one easy window. Now, instead of using the FASTQ files as your starting point, many of you actually, are interested in starting from a matrix count that you obtained outside of OmicSoft. And so in OmicSoft, you can analyze your matrix data as well. And so for those of you that are following along, today we're going to be working with this matrix. This is the counts matrix. And so here, the first column just has the genes that were quantified, and then the subsequent columns represent the samples coming from 2 cell lines. And so here are the values represent raw counts. Now, some of you may be working with normalized counts as well. So we will be working with this matrix as well, where we have the genes on the first column, and then the samples in the subsequent columns from 2 cell lines. And so here, the values are going to represent normalized counts. And then lastly, we're also going to be working with what we call a design table. And so this design table basically represents the metadata or descriptive information of your data. This table provides some right sample level information that you will need to perform any type of grouping for your analysis, whether you are starting from FASTQ files or from a matrix. Now, in terms of formats, you do need some sort of ID, which is your sample ID or your observation ID that matches your matrix files. And so here, for example, the observations in the matrix are going to match those in the samples, right? So we do have some matching going on there. And then the subsequent columns just give you some sample level information. So here we can see, what tissues the samples are coming from, what cell lines, and so forth. And so here you can see that we're working with cell lines that are originating from bone marrow and breast tissues. These are the files that we are going to be working with, and these are the ones that are provided in the slides for today. So to start off, we need to import your matrix files. And so to do that, I'm going to go ahead and highlight the demo folder, since it's the one that we're going to be working with, and go into the Add Data option. And under Add Data, we're going to go ahead and select the option to add Omic data. Now, in this case, we're going to be adding expression data. So we're going to go ahead and select that. So this is going to allow you to choose the structure of your matrix data. And so in this case, we are working with an Excel spreadsheet where we have the variables as listed in each row. And so you're going to use the option Excel file variable per row. In this case, we're going to go ahead and click on OK. And then we're going to go ahead and import our count table. So selecting the count table, clicking on Open, right? And then because we are working with an Excel spreadsheet, we're going to choose the first sheet and then go ahead and click OK. So here you can see the samples that we're going to be working with. So we're just going to go ahead and click OK. And so once you import the matrix, a window pops up that will ask you if you want to attach a design table. So in this case, we can go ahead and click on Yes and select the file format that the design table is in, which is going to be an Excel file in our case. Click on OK and select that design table from your local computer. Again, we're going to be working with spreadsheet 1, so we're going to go ahead and click OK. And here under Specify Options, we're just going to click OK. Now, once you add the design table, it's going to ask you if you want to add any annotations. We do have annotations, so we'll go ahead and click on Yes. Our annotations table is in the Excel file. And then we can go ahead and select our annotation table. Look at the sheet 1 within that spreadsheet, and then click OK. So once you do this, the first thing that you will notice is that you now have some new items to look at. So here, when we take a look at our folder, we now have an Omics data object where we have our object that we just added, right? And so if we take a look at this Omic data object, this represents our raw counts coming from our matrix. So let's go ahead and rename it so that we know what type of information this is by simply highlighting it, doing a right click, and click on the option to rename. This was our counts. This is a little bit more descriptive of what that file is. And then you can see that we have an annotation and a design folder as well. So here, if you click on the table, that is going to represent this table that is currently opened. Now, if we open up these folders, we can take a look at the design table by clicking on the table and taking a look at what that metadata that we uploaded looks like. And then if we go under annotations, we can take a look at that table for the annotations to see what annotations we uploaded, right? And so here you can easily see that you've uploaded your first matrix. Now, since we are also working with normalized counts, we can go ahead and add those as well. And it's going to be the same process that we did with the raw counts. We're going to go ahead and go into the Add Data, Add Omic Data, Add Expression Data, and then here we're going to select the Excel variable per row, select our table, look at it from the first spreadsheet, click OK, and then we're going to add a design table It's going to be an Excel spreadsheet. So we're going to click and select that design table. We also have annotations associated with that. So we're going to select those annotations from the Excel file and then select the annotations for that corresponding table. And so this is going to add another object. And so here we can just rename it to represent what it is. Now we have our FPKM and our raw count table. And so with this, let's go ahead and take a Q&A break to answer any questions that have arisen.
Wonderful. Thank you so much, Araceli. All right. So a few questions have come up. So one of them was, can OmicSoft actually interface with AWS as the server?
Sorry, can you repeat that? My audio just cut.
Oh, sure. Can OmicSoft interface with, say, AWS for server?
Yes, it can. So we do have the ability to use AWS interface within the server, OmicSoft. So that is a possibility.
Wonderful. And then let's see. One of the questions was about changing colors and shapes in some of the interfaces there, or some of the images there. So if you don't mind, just showing those off when you get to those steps there, that would be wonderful. Another question that came up is, what are some of the resources that are available for brand spanking new users to get started with OmicSoft?
That's a great question. So if we go under help, we do have this option called Getting Started. And so this is going to open up a page in your browser that's going to provide you with our onboarding portal. And so here, if you're a brand new user, you can take a look at the different options that are available for you. So this really allows you to, browse in and take a look at how you can use the OmicSoft software to look at different types of analysis, different types of topics. So that is available by just simply going to the help and then getting started.
Excellent. Thank you so much for that. Another question that just popped up is, what is the difference between raw counts and FPKM?
So the raw counts are going to be, your counts that you get after mapping your genes to reference. The FPKM counts are normalized. These are normalized counts. That's the main difference. One file is normalized counts, and then the other file is not normalized.
Excellent. All right. So no other questions in the Q&A box. So you guys out there in the audience, please keep those questions coming so we can answer them throughout the webinar. And I'm going to go ahead and paste today's slides in chat. And remember, Araceli has wonderfully made some step-by-step guides. So whether you guys are running on a local instance of OmicSoft for your analysis or server instance, there are step-by-step guides for that inside of those slides that I just shared. So Araceli, I'll give it back to you.
Awesome. Thanks everyone for the slides. And then based on the speed poll, it looks like the pace is just fine. So that's good to know. Okay. So now that you have your data uploaded and ready to go in OmicSoft, before doing any expression analysis, you may want to look for any variability in your data sets. And so a principal component analysis can be used to detect outliers and to get a general idea of the distribution of your data. And so to run a PCA on your data set, you can simply go to the option up here for Omic data. So I'm just going to go ahead and just close this file so that everything looks nice and clean. But up here, these different menus provide different tools that can be used for the different types of analysis that you're interested in. And so here we use the Omic data menu. You can see that we have different types of tools that are available to do, for example, QC, pre-processing, and so forth. Now here, we are interested in doing a QC because we want to take a look and generate a PCA. And so under the QC section, you will see the option to do a principal component analysis. And so what OmicSoft is going to do, it is going to automatically select information based on the last project that you opened. And so here, if we take a look at the project section, it is looking at the demo folder because that's the one that's currently highlighted in the Explorer. And so if you have multiple projects that you have opened in the solution, you want to make sure that you are looking at the correct project. And in this case, OmicSoft will also select the data format for that particular tool. And so in this case, it is selecting the FPKM. So just want to a key thing to note is that, if you have multiple projects within your navigation area, you want to make sure that you're looking at the correct project. So here we're going to be using our FPKM, our normalized counts. And so you can then add what type of analysis you want to perform, whether you want to take a look at all of your genes or maybe certain genes, all of your samples or certain samples. And then here under other options, you can select the number of components that you want to analyze for your PCA. And so in this case, we can select that we want to do 3 components. You can also group your samples by any metadata that you provided. For our case, we're just going to use none, but you can see how the metadata can be very useful to help organize your analysis. And then lastly, just select the name of the output. So I'm just going to go PCA plot.
Sally, just to interrupt you real quick, it doesn't seem like the pop-up is being shown on your screen. Do you mind just resharing your full screen there so folks can see that pop? There it is. Awesome. Thank you.
Awesome.
Thank you.
All right. So I apologize about that. So if the pop-up didn't show, just to get that, you go under Omic Data and then QC, Principal Analysis. You do want to make sure that you're looking at the right folder. You have multiple open. And OmicSoft will select the data type that it's going to look at, in this case, the FPKM. You can take a look at, whether you want to look at all of your genes, all of your samples. And here under the option section, we can change the number of components that we're interested in to, let's do 3. You can group your data based on your metadata, so you can see how that can be very useful. In this case, we'll just leave it as none. And then lastly, you can name your plot. So I'll just do PCA plot and then submit that information. And so OmicSoft will generate that PCA plot for you. So once the PCA plot is generated, it is going to create a new folder here under the table section. So under table section, we do have this now new QC folder where we have our PCA output. And so here you can see the PCA. And so this is a interactive plot, right? So you can move it around and play with it. If we go under your view controller and click on the option here under change symbols properties, you can customize it a little bit better. You can change the colors, make it however you want. So in this case, if we select that option, we can specify what we want to color by. So let's say that we want to do a coloring by the cell line, and this is going to be a categorical. So you can see that now we have our one cell line colored in green and then the other cell line colored in blue. You can also play around with the size, and then you can also play around with the shape. So if you wanted to shape by, let's say, tissue, you can add that information as well. right? So when you close this, you do have a more customizable plot here, and you can see what that is in the legend. And so taking a look at this, the goal here is to see what variability is present in your data. And so you can see that component 1 accounts for most of the variability. We also see that one group of samples segregates to the right, the other one segregates to the left. And then you can see that, there is some clustering of some samples, and there's some outliers within those different groups. And so again, this allows you to take a look at, how your data is similar or different between each other. Now, just a show of hands, how many of you are interested in generating an output like this, where you can easily see how your things cluster? I can see that many of you are interested in that. Now, once you take a look at the QC, right, many of you will also want to take a look at how your genes are differentially expressed. And so I work with RNA-Seq data. Many users like you typically are interested in differential expression data. And so in OmicSoft, you can use the count matrix to look for differential expression. And so we can calculate differential expression by using the tools that are available within the NGS menu. And so here under the NGS menu, if we take a look at the inference section, we offer different statistical tests that you can perform to calculate your differential expression. And so today we are using a very simple data set involving 2 cell lines. So we are actually going to be using the DSeq one-way test. And so this test offers a statistical method to generate comparisons between two groups of samples, right? Now, if you have more complex groupings, or you want to do a more complex comparison, you can always use the for example, a DSeq general linear model that allows you to better define the types of comparisons that you want to generate. But because today we're using a very simple one, we're just looking at 2 different things, we're going to go ahead and use the one-way test. And so this is going to open up the tool. Again, you want to make sure that you are working with the same folder, right? So if you have multiple folders, you do want to make sure that that folder is selected under the projects. And then you can see that under the data, it has selected the raw counts as the appropriate type of data that we're going to be looking at. Again, under variables, you can select what particular genes you want to focus on. Under observations, you can select what particular samples you want to work with. And then here under the options section, you can specify how you want to generate those comparisons. Now in this case, under grouping, we're going to be grouping by cell line. And then because we don't really have a true control in terms of the comparison, we're going to do a pairwise comparison. And then lastly, you can specify what type of multiplicity you're interested in, and then your significance level and so forth. So here we do provide you with different options in terms of what type of output you want to generate. So in this case, if you don't want to do a dispersion table, you can deselect that option and so forth. So once you select those options, you can just simply name your output and submit that information. And so this is going to calculate that differential expression. And you will notice if you go within your table section that you now have a inference folder that has been added. And so the inference folder is going to contain the outputs for your differential expression. And so the differential expression outputs within the inference folder is going to include a report, which represents the table that you're looking at here. And it's also going to generate a visual report, a visual output, which is going to be the volcano plot. So since we have that table open. Let's go ahead and check it out. And so here, this table is going to give you every gene, and then take a look at differential expression. So here you can see the fold change between cell line 1 versus cell line 2, the p-values, the FDR values, and so forth. And so here, one of the things that you may want to do is you can, filter this table for significance. And so the filtering options can be accessed by going into the task tab here, and then using the column and the row to filter based on specific fields within this table. Or you can just simply utilize the little filter icon that's located up here. And so this filter icon up here is going to open up this pop-up that allows you to pre-filter for specific things. So in this case, you can filter this table for significance. You can go under the fold change filter and type in, what type of fold change you want to filter for. So let's say that you want to filter for, a absolute value of 2, right? So you can do filter for greater than or less than -2, and click on enter to apply that filter. You can also, for example, filter by p-value. So you can enter a value, let's say, of less than 0.05. So we can go ahead and enter that. And then again, click on enter to type that in. As you can see, as we are filtering, you can see that our number of rows are being filtered to only those that specify those requirements that we are interested in. Now, in addition to generating differential expression data, many users like you want to further analyze that data within a biological context. And so QIAGEN's engineering pathway analysis is a very powerful tool that allows you to perform analysis interpretation of different types of omic data. Now, by using the different features within IPA, powered by its knowledge base with over 13 million manually curated literature-based findings, you can upload your differential expression table to generate different visual outputs to gain further insights into the molecular mechanisms behind your data set. So you can easily see how this table of differentially expressed genes is associated with different pathways. And you can take a look at, the enrichment and the predicted behavior of those pathways by looking at the colors, where predicted activation is represented by orange and predicted inhibition is represented by blue. You can take a look at different networks to see how your genes found within those pathways interact with each other and with downstream functions. And then you can also identify different targets and see how they are expressed across different conditions or different types of omic data. And so OmicSoft is connected to engineering pathway analysis, right? And so here in OmicSoft, you can easily upload your differential expression data directly into engineering pathway analysis by simply going to the integration and then selecting IPA and then upload. And so here you can choose your differential expression table, which is highlighted automatically because we were looking at it in the Solution Explorer. You can select your comparisons. Again, these are going to be automatically selected because that's the data that we're looking at. And then under the Analysis tab, you can choose the option to generate a core analysis and set your cutoffs and what's not, and then submit that information into IPA. And so this will send your differential expression table to IPA, where it will generate a core analysis for you that you can easily view once you're in that software. So for those of you that are interested or have access to IPA, do note that you can send your data that you generate from OmicSoft directly into IPA by simply going to Integration, IPA, and then Data Upload. Now, going back to our outputs that were generated for the differential expression, it generated a table, but as I mentioned, it also generates a volcano plot. So if we go into our Solution Explorer, double click on that, you can see that it generates a volcano plot. So the volcano plot is interactive, so it interacts with the table. So if you make any changes on the table, they are represented in the volcano plot, and that's why you can see that there's some very distinct cutoffs already set in this volcano plot because the table was filtered for significance. And so here, this volcano plot can be customized to fit your visual needs. And so by utilizing the information within the Task tab. So here under the Task tab, we can specify some cutoff lines by using the option Specify Cutoff Lines. And so if we go ahead and just click OK, we can see the lines for particular cutoffs being specified. You can also change the way the volcano plot is colored, the size of it, by simply going under the Task tab and then going under Properties and then selecting the option Change Symbols Properties. So this option is going to just kind of know that this is that. So this is going to allow you to specify, coloring. So here under Color, if we do a continuous color and use the drop-down to select full change, for example, we can color based on full change. Under size, you can increase the point size to make them really big, really small, until you find a size that you're interested in. And so once you close this window, you can see that you've made those different adjustments. And so here, this is possible by simply, again, going to the Task tab and selecting the Change Symbols properties. And so you can see here now we have a table, a volcano, where you can see the red is upregulated and then the green represents downregulated, which is represented here by this range. You can always adjust the coloring of the volcano plot by just simply right-clicking on the range to change the color. And so here, we may remove that black to white. Oh, guess it did. There we go. There we go. And now you can see it a little bit better. So this allows you to definitely customize your outputs. And so, as I mentioned, this is interactive. So if you want to know what some of these highly upregulated genes are, you can select them on the volcano plot, and it's going to open up details at the bottom. And then you can also select some data points from the table and then take a look at them in the volcano plot as well. And so here we can see, that, for example, CCND1 is highly upregulated. This gene is involved in cell cycle. So with this, we'll go ahead and take another Q&A break and answer any additional questions that came in.
Excellent. Thank you so much, Araceli, for this wonderful presentation. Lots of good stuff in it. All right, so some of the other questions that came up was, how can we export some of these images to include them in, say, a publication?
So there's different export options that are available. So you can export this as a graphic by simply just clicking on the little export icon. So here, you can export this as a graphic. You can also export this into a PowerPoint slide by clicking on the little PowerPoint icon here and then selecting, to open it in PowerPoint. It'll do that as well. So we do have different exporting options that are available for you to look at. So this is what the export will look like. Excellent.
Another question that came up was somebody was wondering if this DE-Seq2 one-way test that you chose, is that the same as DE-Seq2 walled T test?
I am not familiar if it is or not.
I believe when you go to inference and the one-way test, I believe you may be able to configure a walled table to be, there you go.
Yes.
There you go.
Yeah.
So if you wanted that walled statistic to be pumped out of there, that's one way to do it right there. So yes and no is the answer to that question. All right. Any other questions? Let me just look here. Nope, no other questions at this time. I am once again going to just paste the slides in chat for anybody that has not grabbed those slides yet. Even if you're not following along today, there's a ton of incredibly useful resources in those slides there. Also, I am going to just check in with the speed. Yep, looks like we are going at a good pace. So thank you guys so much. And if you guys have any questions so far, please definitely submit those to the Q&A box here, and we can get to them during the last Q&A break here. So with that, I will hand it back over to you, Araceli.
Awesome. So thanks everyone for typing in those questions. And again, if you guys have any other ones, continue to type them in the Q&A box, and we will answer them as we go along or towards the end. Now, going back to this differential expression table, right? Let's say that you're interested in generating a list of the top significantly differentially expressed genes, and you want to use this list to generate a heat map to prioritize your target genes. Now, you can easily do that by sorting this table for expression, right? So if we go to the fold change column, we can do a right click and sort by either a ascending or descending. We can do that same thing with p-value. So let's go ahead and do that in the p-value, right? So here p-value, we can do a sort ascending to take a look at the top significantly differentially expressed genes. And we can go ahead and select these to create a table, or sorry, not create a table, create a list. So in this case, I'm going to go ahead and just select a few of these genes just to kind of, get some numbers. So we'll go ahead and select these. And so you can see that they are highlighted in bold. And so once we select the genes that we're interested in, we can create a list out of these genes by simply going to the option of add item. And here under add item, we're going to add a list from selected rows. And so again, we went to add item and then add item from selected rows. And then this little window allows you to select, what option you want to use. In this case, we want to use the gene ID, the first column in this table. And then we can go ahead and name it. So I'm just going to put top significant genes and then click OK. So now you can see that we have our table listed right here. And so we can use that table to go ahead and make our heat map. So if we double click on it, we can see what those top significant genes are. And so using this list, we can generate a heat map and perform a hierarchical clustering analysis by going into our Omic data menu and then selecting the option for pattern and then hierarchical clustering. So again, Omic data, pattern, and then hierarchical clustering. And so here, we're going to go ahead and select, make sure that we're working with the right folder. And then in terms of the data, we're going to use the normalized counts. And under variables, we do have a specified set number of genes that we want to work with. So we're going to use the customize variables option. And then under select, we're going to go ahead and select our list. So instead of looking at the 30,000 genes or transcripts, we're just going to focus on 153. Under observations, we do want to look at all of our samples, so we'll leave that as is. And then under options, you have the option to compute an observation tree and a variable tree. So let's go ahead and select both. And then in terms of the length and the distance, we're just going to leave them as default. And so lastly, we can go ahead and just name our output. And submit. And so what this is going to do, this is going to generate two types of heat maps. So if we go into our Omic data object and within our FPKM, we can see that we have our two heat maps listed, and they're also going to be displayed here on your window. So I'm just going to go ahead and just close a couple of things just so that the window doesn't get crowded, and that way you get to see, what output is which. And so the first output that is going to generate is going to generate this non-classical heat map that gives you a gene breakdown. So here we have every single gene, and then you can see the samples listed right here. Now, the second one that most people are used to seeing is what we refer to as the classical heat map. And so here we have a heat map where we have our genes on the y-axis and then our samples on the x-axis. And so you can customize the way that this heat map looks by simply going under the Task tab and then going into the different categories that are available within the Task tab. So let's say right now we have this color label that is coloring what the tissues are, and you can see up here that we're looking at bone marrow and breast. Let's say that we also wanted to add a label for our cell lines. We can easily do that by going into the task, and then under the customize section, we can select the option to change x-axis color bars. And so using that option, we can specify to add a color bar that also gives you information on the cell line. Click OK. And so now we have two different color bars. We can also we can also change the color properties of this heat map. So if we go under task and then property, change color properties, we can say, okay, let's get rid of the black coloring. It makes it a little too dark. Let's switch that to white. And that makes it a little bit better, right? And then you can also change the coloring by going into the legend here, right? So if you want to change the way that the color bar for bone marrow and breast is represented, you can highlight the name, right click, and change color to a different color. Let's say that you want to color this orange. We can do so, and then we can change the color for the breast tissue to color this, let's say, purple, and we can customize the input of how that heat map looks. And so here you can see that the set of genes are highly expressed in the bone marrow tissue, whereas these set of genes are highly expressed in the breast tissue. And this allows you to identify some target genes that you can take a look at in more greater details. Now, let's say that you have your list of differential expressed genes, and you find a key gene that you're interested in, right? And so what you want to do is you want to take a look at, at the gene expression, but you want to take a look at expression, but as at the gene level. And so one of the things that you can do is you can generate gene level expression plots. And so since we're looking at normalized counts, we can add a view to take a look at that normalized expression by simply selecting our object and then doing a right click and adding a view. So here we have different types of views that you can add, but the one that we are interested in is looking at a variable view. So we can go ahead and select the variable view and click OK. And so what this is going to do, this is going to generate charts for each of the individual genes across your different samples, right? So here we're looking at the chart for this particular gene. And so it's kind of hard to tell, exactly which samples belong to a specific cell line. So we can easily group these samples together by going into the Task tab and then using the option Specify Profile Columns to sort of resort these charts. So rather than sorting them by the sample, let's go ahead and sort them by the cell line. And so now we can easily see our samples that belong to one cell line versus our samples that belong to a different cell line. We can also color the samples by cell lines by, again, going to the Task tab and then going under Properties and change Symbols Properties. And so here we can do a color, and then we could do a categorical color by cell line. So now our cell line samples are in blue, and then we also have some in green. And so here you can take a look at how different genes are expressed across, those different samples. Now, because, we have all these different tables and we would rather focus on our gene of interest, you can filter for your gene of interest by simply utilizing the variable tab here. And then you can click on the filter to filter by gene ID, for example. So if we go back and we focus on that gene that was highly differentially expressed, which was CCND1, we can take a look at that table that's specifically for our gene of interest. You can see that is highly expressed in the MCF7 cell line compared to the K562 cell line. And so this is one way to visualize it, but let's say that you want to change the way that this chart looks and you're interested more in box plots rather than the scatter plot. You can change that by simply going into the Task tab, and then here in the Task tab, you can go under Customize, and then change Profile Gallery. And so this will allow you to change the way that the chart looks. So we can go and do like an R box plot style or just a box plot, and then visualize it this way. And so if you want to take a look at some summary statistics, you can go into the Task tab, Customize, and then show summary information to get that those statistics listed here. And so this is one way to take a look at how your genes of interest are expressed across your different samples. So just a show of hands, how many of you like the idea of being able to change the way that your charts look to give you a more visually pleasing way to take a look at that analysis? So I can see that many of you find that a great option to have. Okay, so some of you are maybe interested in a gene set analysis, right? And so gene set analysis is a powerful tool that can help users like you who have their own gene set and would like to identify comparisons containing similar gene set enrichments coming from tens of thousands of comparisons from the public domain. And so in OmicSoft, you can perform such analysis by using the gene set analysis option within the GUI. And so in OmicSoft, you can perform a gene set analysis by going to integration and then gene set analysis. However, this option is only available for server projects. So you need to make sure that you are working with a server project and not a local project before performing this analysis. So this is kind of like the only difference when it comes to the type of project that you use. All the tools will work on either projects, but this one tool, the gene set analysis tool, it is server project specific. And so if you want to use this, you need to make sure that you're working with a server project. So if I highlight my little server project here, you can then use the gene set analysis tool. And so if you don't believe me, if I select my demo folder, which is my local, go to integration, gene set, it will let you know that you do need a server project. So just to make sure that you're working with the server project, if your intentions is to generate a gene set analysis as part of your analytical steps. And so the gene set analysis is available again through integration and then gene set analysis. And so here you can select the universal set name, which we do have different options to choose from. And then you can also select which databases would you like to search in. And so these are databases coming from different sources, including the OmicSoft lands that we curate as well. So let's say, I select this one as an example. You can also define the different list that you may have, right? So under the what type of data you have, you can say, I have a list of identifiers. I have a list with some directional expression and so forth. So you can specify what type of information you're going to input, and then also what IDs you're going to use. So once you select your options, the next step is to upload that information, right? So previously I noted that I'm working with the list. So you can upload your list. Let's say that I'm using this list. You can see the list of genes that you're going to be working with. So you can upload them from your solution, or you can also do a local upload as well. Once you have your list, you can then select how you want to set up the analysis parameters. And then lastly, it's just going to let you know what genes it was able to identify and which ones it was not able to. And then you can just click on Finish to run your gene set analysis. So this actually is a little bit of time consuming. So I'm going to go ahead and click on Cancel. And I already did this ahead of time. So I'm going to go ahead and actually open the server project in which this was done. And so when you run and finish your analysis, it's going to generate different outputs, different reports that allow you to identify, what were the top comparisons that have your list of genes. And so you're able to explore that information by taking a look at the different options that are available and then the different studies within it. So just a show of hands for those of you that were not aware, that were familiar with OmicSoft and were not aware that this was an option, let us know. So if you didn't know that you are able to do a gene set analysis within OmicSoft, it's great to kind of spread the word to make sure that if you're using OmicSoft, you know that these tools are there. Okay, awesome. And so today we took a look at how you can analyze your data for bulk RNA-Seq, either starting from FASTQ files or from a matrix by utilizing the Array Studio through a project. So if you are starting your analysis from FASTQ files, you can analyze your data individually by running individual steps or by using a RNA-Seq pipeline to do the analysis all in one step. Now, if you're starting your analysis from a matrix, you can add that matrix into OmicSoft by simply just importing it in. And then once you have your data uploaded, you can run different analysis. So you can do a PCA plot to take a look at the outliers that are present within your data. You can do a differential expression analysis by selecting the statistical test that is appropriate for you. You can generate heat maps and volcano plots, and you can also generate views for looking at single expression for individual genes. And so with this, thank you everyone for taking time to attend today's session. And again, if you guys have any questions, we'd be happy to take them. And do, if you have time, fill out that survey. It lets us know how we're doing, what topics you're interested in. And this gives us an idea of, what other use cases to present in our webinars that we do on a regular basis. So with that, Kristen, if there's any questions, I'd be happy to take them.
Excellent. Thank you so much, Araceli. This was awesome. So first things first, I did share the link to the post-training survey. So if you guys want to say how absolutely spectacular Araceli is at showing you how to do all sorts of things analysis in OmicSoft, please feel free to do so. We love hearing your feedback. So one of the questions that came up actually was around that GSEA. So they were interested to know that you can actually run GSEA in OmicSoft. They thought that you'd only be able to do that with IPA. So maybe if you could just kind of briefly go over what are the differences between GSEA in OmicSoft versus what you'd be able to do in IPA?
So a GSEA in OmicSoft is basically going to tell you about enrichment, right? Where those genes have been noted, right? But the key difference between OmicSoft and gene path analysis is that in addition to telling you where your genes have been looked at, IP is going to tell you how those genes affect behavior, right? And so this is going to allow you to take a look at how that gene expression that you're interested in for those particular genes are influencing the activation or inhibition of different pathways, the activation or inhibition of different diseases, different functions, different key regulatory molecules that are involved in your data. So that's the big difference. The GCA and OmicSoft just basically tells you enrichment, here are my genes, this is where they've been observed. But IPA will tell you a lot more in terms of biological context.
Fabulous. Thank you so much, Araceli. Another question that came up was around that heat map that you generated. So it looks like you generated that heat map and we were looking at the FPKM values for all of the different samples that we brought into our comparison there. Would you be able to generate, say, a heat map of the fold changes if that's something that you wanted to do in OmicSoft?
Yes, so you could do of the fold changes. So right now we're doing actual expression. I don't know why it's not showing, but yes, you can generate a heat map based on fold changes as well. And so the way to do so is by generating that list. And instead of focusing on the gene ID, you can focus on the fold changes and you can generate the heat map that way.
Fabulous. It looks like OmicSoft is starting to get tired. It's getting towards the end of the day here, so I understand that everybody needs a break, right?
Yeah.
Someone just in the Q&A box was asking, can you analyze Olink data with OmicSoft? Interesting question.
I'm not familiar with Olink.
Yeah, Olink is one of the protein platforms that does like massive amounts of proteins in one fell swoop. So unfortunately, we don't have tools for that in OmicSoft, but of course, wait a minute and things could change. That's something that I can definitely share with the product manager to let them know that there is interest in that. I do know that in the future, we will start including a lot more proteomics data, including Olink generated data in our lands. So I imagine that the same analysis tools that were used for those lands data, maybe we could get them, accessible to you guys through our analysis tab. But thank you so much for that feedback, and I will certainly let the product manager know that there is certainly interest in that.
Well, that's great to know.
Yeah, pretty cool. Yeah, Olink is super cool because you can look at so many different things all at once instead of some of these older protein array kind of things. If you guys could just fill out that final meeting poll question that popped up, that would be wonderful. And I'll hang out and wait to see, do we have any more, straggler questions here? We'll just chill for a few moments here. So thank you guys so much for hanging out with us today. Please fill out that post-training survey if you have a moment there, sharing that link again in chat. And then just for good measure, I will share again today's slides in chat. Remember, there are some wonderful step-by-step slides that Araceli has graciously constructed and shared in that slide deck. So thank you guys so much. Thank you again, Araceli, for being such a fabulous teacher. And we hope to see you in future webinars. We'll hang out for another minute or so to see if we have any last minute questions.
There's a question in the chat that says, how did you upload the data onto the server? So
yes,
if we go onto the server, you can just simply upload data by clicking on the upload. So here under upload, you can select what file you want to upload. So let's say I want to upload this design table. Select it, and then it will be uploaded onto the server.
Perfect. Yep, and I've linked, and Araceli's also linked those step-by-step slides to kind of bring you through this process as well. So feel free to tap into those. And then, of course, at the end of the day, if you're still like, man, I don't remember anything from this webinar, just reach out to us for one-on-ones. We're more than happy to help you out. Okay, so it looks like we have no other questions. So thank you guys so much for hanging out with us today, and we'll see you in future webinars.
QIAGEN.
Sample to insight.